Hello and welcome to this discussion about molecular and genetic epidemiology. This is a very short introduction, and I want to stress that this is a ginormic field. In a lot of universities, they have either a whole entire class in just genetic epidemiology, or they may even have a whole degree in genetic epidemiology. So you're getting a tiny little taste of something that is much bigger, and hopefully this will help frame some of your ideas about how diseases in populations can be studied. So as a starting point, I like to think about the person, and your book actually talks about this like a black box, so I'm going to draw it as a black box. A person, and a lot of times people are influenced by environmental factors, and then we see some kind of disease manifest itself. And these exposures here really could be anything. It could be medications that they take, it could be lifestyle habits that they have, it could be any kind of thing that is part of the causal pathway that helps contribute to a person having that disease. And a lot of times it's important to remember that there are multiple things that have to all be happening at the same time for a given person. We'll give this person green eyes. Say they're happy. So there are a lot of things that need to all be happening at the same time in order for that particular person to end up having the disease that we're interested in studying. Now, this black box has to do with everything that's happening inside of a person's physiology. It includes their DNA, it includes all of the proteins and molecules and cells and everything that happens inside of the person's body. So, in reality, these causes that we think about could even be things that are within the person's body that sets them up for being more at risk, and we'll talk about that when we talk about the genetic epidemiology. So this whole idea of molecular and genetic epidemiology could be that we're looking for the places where this exposure, let's say somebody's exposed to lead paint, or they're exposed to certain water or air or certain medications or just whatever it is, we're looking for markers in the person's body. So this could be one of the biological things that we're looking for. So biomarkers of how much exposure a person had. Now, the interesting thing about biomarkers is that two people could have the same exact exposure to lead or a certain food, but the biomarker that we collect is different for that person. And so they have accumulated more of a certain kind of fatty acid, or they produce more of a kind of protein in response to that. And so what biomarkers does is it allows us to take into account that individual response is different for different people. So not only are we looking for biomarkers, we're looking at a person's individual response. And the other thing a lot of times that we're doing inside of this black box is we're looking for, as this process is happening, let me see if I can just draw it in green. As this process is happening from the exposure to the disease, along that pathway, a person isn't just instantly have like stage 4 cancer. It's a process that is constantly going inside of a person's body. So we can look at different precursors. So maybe they have early cell changes here, and then later on it starts forming more of an area, and then maybe they have stage 1 cancer, and by the time they become symptomatic over here, it's gone through a whole process. So we can look at precursors. And so this whole idea of molecular and genetic epidemiology is looking into the black box. We're making it transparent so that we have more of an idea of what's happening for that particular person. So I have broken this down, as your textbook has, into the genetic and the molecular epidemiology as a 20,000 foot view of how these are the same or different. Genetic epidemiology is about things that you inherit specifically from your parents. And so your biological parents, you got half of your genes from your mother and half of your genes from your biological father. And those inherited factors determine a lot of these things in terms of how you respond to disease, what are even the things that predispose you to being more sensitive to certain things, and what happens is, if this is the kind of inherited factor that clusters in families, and we call that Mendelian transmission. There's a certain, your textbook talks about that if you're not familiar with it, but it's how genes are passed from parent to child. So if you look at the bottom of page 606, the genetic epidemiology is a collection of methodologies that, first of all, at least the historical kind of genetic epidemiology, is we're asking the question, does this disease of interest cluster in families? And so that is a very key feature of genetic epidemiology, because a lot of times with genetic epidemiology, what happens is we're looking for this cluster as a starting point. We don't just say, okay, there's a person who has the disease. We want to know off the bat, is this clustering in the family? And so what we need to do is take a family history and we say, okay, do you have anyone in your family who has this disease or that disease or all these different kinds of things? And there are a lot of complications about asking questions like that, as your book talks about. But the bottom line with genetic epidemiology is, first of all, we need to see if it's clustering in families. So we're looking at that inherited factor that influences disease or risk of disease. And we're looking for an excess occurrence in a specific family. Now, that can play out in terms of how that variation in genetic material actually interacts with the environmental factors. So a person can have certain genes, and that genetic difference in that particular family means that they are either more or less sensitive to some kind of an environmental factor of interest. It could increase the risk of disease or even theoretically decrease the risk of disease if it's protective. And so, first of all, if we know about this whole idea that it's clustering in this particular family, that can give us information that would help us detect this earlier in that particular family. We say, okay, you have that gene, and so you're high risk, and so we want to find it early for you. But also, what this does, finding out about these genetic differences, gives us the opportunity to design more effective interventions. So not only are we finding it early for that particular person, but it gives us a clue about the biology of how this factor, this environmental influence, is causing disease. So this intervention idea. So it gives us ideas about what could help, sometimes not only for the group that has this genetic difference, but even for other groups. So a key feature here is that we need to have a person's relatives. And what that does is it makes this into a study design, where those individuals are not independent from each other. And so let's say we have somebody, Sally, and her brother, Joe, and we find out that both of them have a particular disease. We take all of their DNA and line it up and look for places on that DNA between the two people that match. And so we're looking for that matching. And when we find places that match between two individuals that are within that family that are having this high incidence of disease, then those are the suspect areas. And genetics is very complicated. There's a lot of matching, because not all genes are determining things about an individual. So it's much more complicated than what I'm drawing here, obviously. But that's the general idea. If they had, say, a third and maybe even a fourth sibling, and those did not have the disease, and you looked at that same area and they did not have something that was matching, that helps us really be able to see what areas stand out for the individuals that do have disease. So that's kind of in a nutshell how we're looking at that. But the main thing to remember is that genetic epidemiology really is this study of families. And we can study a variety of families and have control families and all of that kind of thing. Now, the trick is that genes are actually a kind of a molecule. And with something called genotyping, we can look for causal genetic variants, these little individual differences right here. We can look for snippets now in individuals who are not related. And so that just because the technology has gotten so much better, we can start looking for those kinds of patterns in people who aren't in the same family. And that's where this whole thing of genetic epidemiology really starts kind of spilling over into molecular epidemiology. So genes are molecules, and genetic epidemiology is a special subset where we're really looking at how these inherited factors cluster in families. And as soon as you're taking that out of the inherited factors or clustering in families, then you're talking about molecular epidemiology. Let me go back really quickly to the bottom of page 606. The first point is that the disease of interest clusters in families. The second point is we can't find another reason why this is happening in this family. Like, it's not just because of their shared lifestyle, smoking, diet, exercise, common environmental things, or similar risk profiles such as water, air, pesticides, lead. Occupation can be a very interesting one. So that having something in common, having it cluster in families does not automatically make it genetic, and so we need to always rule out those kinds of things that could also be causing it. Number 3 on page 606 says, is the pattern of disease within families consistent with the expectations under Mendelian transmission of a major gene? So that's a big mouthful. But in other words, within that family, is it transmitting the way we would expect? And so if it's a dominant or a recessive gene, is it transmitting in the way that Mendel described? And then fourth, then we start looking for this specific snippet on the gene. Where on the chromosome is this gene that matches for all these people in this family who are having that particular disease? Where are the matches? And so we look for those. So that's kind of the process. With molecular epidemiology, then, what we're looking at is a lot of times what we're finding is biomarkers. And by biomarkers, what I mean is this ability to measure something inside of a person. So that measured thing could be that we take a little fat sample from them. Sometimes it's blood samples. Sometimes on rare occasions, we could even be looking at something like bone marrow or muscle or different kinds of things like that. But these biomarkers, things like serum levels of micronutrients, not only do they tell us, give us a more precise estimate of how much a person was exposed to that, it also gives us a little bit of a clue about how that specific person processes that material. Like, let's say you're thinking about measuring a person's, this is an example from the book, how many fruits and vegetables do they eat? Well, we can get a pretty good idea of how many by certain biomarkers that we can measure in a person's blood, micronutrients. However, two people could eat exactly the same amount of fruits and vegetables, and their biomarkers would be higher or lower than each other. So it also is measuring not just how much do you eat, but what is your individual response to that particular exposure. So we could also be looking at sort of this intermediate with the molecular epidemiology. We could be looking at intermediate outcomes. In specific cases, we will look at a person's biologic makeup, and we will say, how is it, now that we know they have a particular disease, can we tailor the treatment for that particular disease? And another important one that you'll see quite a bit of is that if we're looking at infectious disease agents, a lot of times we can tell if microorganisms came from the same strain. They have what's called a DNA fingerprint. And so you have this infectious agent, you can say, well, those two bugs are really offspring of the same infection. Or if they have a different DNA fingerprint, you can say, well, I think they came from different places. They're not just one outbreak, there are multiple different ones. And so if they're the same bacterial strain, that could tell you something about how that disease was transmitted, if they had a common or an identical source, or if they had multiple different sources. So very often with infectious disease outbreak investigation, we're looking at things like that. So we can also, as part of this molecular epidemiology, we can measure inherited variations in DNA as the exposure. So that's really, I'm going to put DNA here as the exposure, so we're pointing still towards the person. That's inside of the black box because it's kind of part of somebody's physiology. But if we can know something about their inherited variation as the exposure variable, then we can know if somebody is more predisposed to having certain diseases or outcomes that are very much related to genetics. Now, the place where molecular biology picks up often and genetic epidemiology stops working very well is where you have multiple genes. And the reason for that is that as soon as you get a whole bunch of multiple genes that are all contributing to a specific disease, then that type of inheritance, the Mendelian transmission of inheritance, tends to sort of break down. We don't see it as cleanly. It's very difficult to pick that out. And the other time is where you have really a kind of a relationship between the DNA and whatever the environmental exposure is, kind of like a confounding or a multiplicative effect. So when you have multiple things that are all influencing it, then it doesn't show up really cleanly as this clustering in families. And so a lot of times molecular genetics will pick up at that point, and genetic epidemiology sort of leaves off. So molecular epidemiology really is much broader and kind of almost encompasses genetic epidemiology, except genetic epidemiology has its own little niche. So molecular epidemiology, usually we're studying individuals. Only occasionally would we really be looking at individuals within a family setting. But here we have the whole idea too of we have environmental exposures. Let me find a new color. So here we have our environmental exposures. Sometimes, and this is another thing that we can look at with molecular epidemiology, sometimes that environmental exposure actually damages the DNA. And so you have this genetic mutation, and that genetic mutation is what causes the disease. And so that damage happens to the person after they've inherited the good gene. It gets damaged and then they progress to have disease. So that's another example of a genetic thing that would not be inherited. So when the genes mutate. Let's see, what else was I going to talk about? So as soon as we're understanding these molecules, then when we add that to the whole concept of epidemiology, that's where we can understand things about the molecular dose, the preclinical effects, the biological factors even that modulate susceptibility to exposure. So what is that individual person's response? And so what this does is it gives us a much more precise measurement. If we're measuring both exposure and disease with biomarkers, remember we're sometimes measuring these precursors, that gives us a much more precise measurement than asking a person a question on a questionnaire, for a variety of different reasons. If you ask a person a question on a questionnaire, they have to remember what they ate, they have to remember how much they exercised, they have to tell you the truth. They can't try to give you the information that they think you want to hear. And it takes into account the whole individual response to that particular exposure. They don't know how their body responds to carrots and broccoli. Maybe they are like a good responder, and they eat a little bit of carrots and broccoli, and it really helps their cells. Maybe they're the kind of person who's resistant to the good effects of carrots and broccoli. There's no way they could tell you that. But if you gather information on these biological measures, you have a much more precise understanding of both the exposure and the outcome. So just asking somebody, do you have this particular disease? They don't necessarily know where they are along that pathway. They don't necessarily know a lot of things about the specific disease. And a lot of times disease has this really wide range. For different people, there are different things that have contributed to it, be it their own personal DNA and the environment and all that combination. So, in summary, I would like to draw your attention to page 640 at the top, and this is where we can really tie in to the previous topic that we had, which was screening. So this paragraph really is talking about an example, and you'll have to read the rest of the example, but basically we're collecting a sample, and I'll start halfway down. Using a panel of markers, they were able to identify correctly 91% of the samples as being derived from cancer cases. Think about, what is that 91%? Let me see if I can draw it here to help you kind of wrap your mind around this. This is the kind of information that you will read in an article or hear on the news, and I want you to be able to think, here I have my screening, I have my exposure, plus, minus, disease, yes, no. So, of those who actually have cancer, look at that table and think, where is that? Of those people who have cancer, this is your yes cancer right here. Of those people who have cancer, 91% of the time they were able to correctly identify them. So here's your 91%. Now we don't know the actual number here and the actual number here. It could be 1,000 or 50 or whatever, but 91% of this total number down here, they were able to correctly identify. What is that? Is that sensitivity? Is that specificity? Is that predictive value of a positive test? Predictive value positive? Predictive value of a negative test? Which one is that? So think about that, and I'll actually give you the answer. It is sensitivity. If a person, of those who have the disease, how sensitive is the test to pick up on that? And 91% of the time, that's the answer. I'm going to skip the middle thing right now. It also says 93% of the samples as being derived from cancer-free controls. So you would have to know that the nose are the cancer-free controls. 93% of the time they were right. So what is that? Of people who don't have cancer, they were right 93% of the time. Let me get a different color. So here's your of people who do not actually have cancer, 93% of the time they were actually correct. So of this, that's your denominator, and whatever number ended up being in here, that calculated out to say 91%, 93%. They also have a middle category of 82% of the samples were correctly identified as being from patients with precursor lesions. So technically, they have kind of a middle category, sort of like we have non-diabetic, pre-diabetic, diabetic. So they've split it into 3 different areas. And so you would have to know a little bit more about how this whole thing is set up in order to do it that way. But I wanted to make sure that it's clear that you can read a sentence like that and you can say, wow, what they're talking about is sensitivity. Of all the people who do have disease, how sensitive was it to pick up on that? And here's your 91%. So that concludes our discussion on molecular and genetic epidemiology. I hope that was helpful.